Daphne Koller is one of the most amazing leaders I've ever met, and we were all delighted that she could give the opening keynote last year at WIDS 2020. Daphne is a Stanford professor, CEO, entrepreneur, and healthcare expert. She is incredibly inspiring with her work at In Vitro using AI for drug discovery. Daphne's keynote here shares the approaches that In Vitro is taking for drug discovery, building a data factory and leveraging machine learning to interpret the data. She emphasized the critical importance of interdisciplinary teams to the success of their solutions and encourages everyone to join the digital biology field.
It's a real pleasure for me to be here and see so many wonderful and talented women here and on the other side of the camera entering a field that I entered almost 30 years ago now in the early 90s, and it makes me feel really old, but and there weren't a lot of women in this field back then, and it's wonderful to see so many wonderful, talented young women entering this space, and I think it'll make a huge difference. So I'll tell you today about what I'm doing in this field right now. I spent my career at Stanford actually working at what I think of as the boundary between two disciplines. One of those is machine learning and data science, and the other one is biology and health. And I use the word boundary rather than intersection deliberately because there really wasn't that much of an intersection between those two fields at the time, because the data that was available on the biology and health side was relatively limited. And so you could apply fairly simple statistical tools, but you couldn't really apply very sophisticated methods. It was a real challenge, and I think that's changing, and I think this is the right time to come back into the space and do amazing things, and I hope that the talk today will be able to convince you of that. So if you think about drug discovery and development, it's kind of a real interesting glass half full, glass half empty perspective. On the one hand, the glass half full is that in the last 50 years, discovery has enabled drugs that have taken diseases that used to be a death sentence or a sentence to a lifelong of pain and agony into something that is now manageable or sometimes even a cure. And cure is a very rare word in medicine today. That includes vaccines for infectious diseases, cancer immunotherapies, autoimmune system modulators. One of my favorite stories is the cystic fibrosis drugs by Vertex, which took something that used to be a death sentence at the age of 20 and now gives 90% of those patients effectively an almost normal life. It is a remarkable tale of achievement. The glass half empty is this other side, which has come to be known as Erums Law. Now, those of you here in the audience, I'm sure are very familiar with Moore's Law. Erum is the inverse of Moore because it is an exponential decrease in productivity of pharmaceutical R&D consistently for the last 70 years. This is a graph on a logarithmic scale where this is the number of drugs approved per $1 billion US. The price tag of approval for a new drug is now $2.5 billion. Now, that is not because that single drug incurs $2.5 billion on its journey from idea to approval. It's because that one drug carries on its back the many, many thousands or tens of thousands of drugs that didn't quite make it. So really what's going on in drug discovery and development is this is a 15-year journey, often, maybe longer, from idea to approval, with many, many forks in the road, where one fork, if we're lucky, takes us to success, 99 don't, and we have only the very limited tools to make a decision on which of the paths in front of us are going to lead us to success, and taking the wrong one can be a matter of years and tens, if not hundreds of millions of dollars before we figure out that we took the wrong turn. So how can we make better predictions of downstream outcomes, hopefully helping bend that curve on the last slide? So this is where I think machine learning and data science could potentially play a role. I'm going to give a very brief introduction to some of the developments in machine learning, maybe not as useful in this audience, but I think interesting nonetheless. I've been amazed at how much progress has been made even in the last 5 years. So back in 2005, I used to do a lot of computer vision, and you would give this image to a computer and ask is there a bear in this image, a simple binary classification problem? And the answer would be slightly better than random. 2012, this is the era of ImageNet, which was created by my colleague here at Stanford, former colleague and friend, Fei Li. And this gave machine learning enough training data to the point that you could actually start answering a much harder question of what is in this image, multi-class classification problem. And the label for this image would probably be wolf. This is actually a bear, but it's a wolfy looking bear, so, you know, not so bad. 2014, an actual label from the state-of-the-art algorithm, a brown bear is swimming in the water. The bear isn't in the water, but there is a brown bear and there is water. So this is actually pretty good. In 2017, 2 bears sitting on top of rocks. Today, the performance of machine learning for this problem is demonstrably beyond human level performance for tasks that each of us has been trained to perform since we were an infant. So the fact that you could actually achieve beyond human level performance is actually, to me, remarkable. Now, what made this possible? So if you look at the models that we used to employ way back when, in the early days of machine learning, these were usually simple models constructed on top of human-constructed features. So things like logistic regression or random forests, and people had to do a lot of what's called feature engineering to feed those algorithms. And it turns out that those algorithms started out pretty good because they introduced a lot of human bias knowledge into this, but they asymptoted at a relatively low level. Today, we basically don't do that anymore. The computer starts out with Scratch and just the raw features, and it takes it longer in some ways. You require more data to reach a high level of performance, but it turns out the computers actually end up constructing better features than people do, which is why the performance keeps going up and up. So specifically, if you look at what we do today, this is what's come to be known as deep learning. And so the computer, for instance, in the case of images, starts with raw features, basic pixels, and it constructs an increasingly complex hierarchy of features built on top of other features, so that in the end, it's able to construct features that are very subtle and can make distinctions like between an Arctic fox and an Eskimo dog, where a person would be really hard put to define a feature that would actually make that distinction. The other aspect of this, which is important and comes back on the next slide, is that there is also a new representation learning aspect here. So if the original images, say, are 1,000 by 1,000, and let's think grayscale for simplicity, then they sit in a 1,000,000-dimensional space. At the end of the day, just before the final prediction is made, that vector is usually about 100 dimensions large. So we have taken 1,000,000 dimensions and compressed it into 100 dimensions, and those are meaningful features that the computer constructed. So from a mathematical perspective, what that actually means is that the machine has actually created a 100-dimensional manifold in a 1,000,000-dimensional space and has embedded all those 1,000,000-dimensional images in that 100-dimensional representation. And that space actually has interesting structure because there's an infinite number, a continuum of manifolds that one could construct, but the machine has to actually put next to each other images that are labeled the same way. So these two trucks, even though in the original space would be as far from each other as any other two random images, here they have to be next to each other. And because the hierarchy of features is learned jointly, other classes that are not labeled the same way but share some features like windshields and tires are going to be next to each other in this manifold, whereas other classes like cats and dogs are in a very different part of the space. Okay, so that's the this is the glass half full side of machine learning. In light of the earlier comments, let me actually talk a little bit about the glass half empty side of machine learning and why quality of data is of paramount importance. So it turns out that these really powerful machine learning algorithms that are really good at teasing apart subtle signal are equally good at latching onto subtle artifacts. So this is one of my favorite examples. It's from an archive paper in 2018. This is a paper that was that looked at how bias can creep into algorithms in the context of a radiology problem where you're trying to diagnose fractures from an X-ray image. This is the input, and you can see they got really good ROC curves. It's, you know, it's not perfect, but really compelling. And then they looked at the embedding of that manifold that I talked about, and they visualized it, and they noticed the following unfortunate fact, which is that if you look at that embedding and you look at the distribution of fracture versus non-fracture, actually kind of nicely distributed across the clusters. So the machine hasn't learned to really distinguish fracture from non-fracture. What it learned to do really well is distinguish the machine, the X-ray machine that took the image. Really, really well. And so it turns out that in fact, when you correct for that, you basically get close to random performance. And what turns out to be the case is that just certain hospitals have a larger predominance of fractures and a certain X-ray machine, and that's what it latched onto, which highlights the importance of having really high quality data as well as a very rigorous testing regime. So we'll come back to that later. Okay, so why am I back in this space after so many years? It's because I think we now have the opportunity to actually create and utilize massive data sets in biomedicine that didn't exist before. So in some ways, there have been two revolutions going on in parallel in two different fields that haven't really spoken to each other. On the left hand is advances in cell biology and bioengineering, each of which has been transformative on its own, but together they've enabled a perfect storm of data creation. So I'm going to talk a little bit about some of these advancements. The first of those is what's called induced pluripotent stem cells. It is an invention, a discovery that earned Yamanaka a Nobel Prize a few years ago. And what it is the ability to take, say, skin cells or white blood cells from any one of us, revert them to stem cell status, which is a pluripotent type of cell that can create any cell lineage in our body, and then differentiate them into neurons or hepatocytes or cardiomyocytes that carry our genetics. And so it enables us to create cellular models that capture the genetic diversity of a normal human population in the cell background that is relevant to disease. We have, using technologies like CRISPR, the ability to furthermore perturb these cells to probe at causality. What would happen if I changed this gene from this form to that form? How does that cell behave differently? We have the ability to gauge behaviors using a range of technologies of high content, what's called phenotyping, measuring those cells using imaging, using super resolution microscopy, using single cell RNA sequencing that measures the transcriptional profile, and many more that are currently emerging. And we can do it all at scale using automation and microfluidics. This creates an incredible amount of data that looks like this image on the left. And people are unable to make sense of that amount of data. And so that's where the machine learning revolution that we're all familiar with can come together with the revolution on the left and really make sense of these data and help us understand biology and find new treatments for disease. So what we're doing is we're trying to put this together, coming back to the Erums Law slide before, to come up with explicitly trained models that can perform predictions on which fork in the road we want to take. So we're looking for places where such a prediction will make a big difference to the probability of success, where machine learning is the right tool for the job, and where we can produce the right data at the right quality and the right scale so that we don't have this example that I showed before with the radiology images. So I can tell you about a number of efforts that we're making along these lines. Since I don't have as much time today, I'm only going to take you about one, but it's the one that we think is the most core, which is how do we predict human clinical outcome? If I make this intervention in a human, what will it do? Now you're sitting here thinking, well, how can you create a data set where you measure human clinical outcome? We don't get to do experiments in people until the very end of the process, which is what's called a randomized clinical trial, and that is absolutely right. And so part of the idea here is how do we use other forms of data to answer this question? So currently, the state of the art, if you will, the normal standard operating procedure for answering the question, what will an intervention do when administered to a human, is, well, we can't administer it to a human, so we're going to administer it to a mouse. Now, the problem, unfortunately, is that mice are just not people. And mice don't get most of the diseases that are the unmet need today. Mice don't get atherosclerosis, they don't get Alzheimer's, they don't get Parkinson's, they don't get autism or schizophrenia or type 2 diabetes, they don't get any of those things. So we create artificial diseases in these animals that are very different to humans, and then we cure these artificial diseases, and then we're surprised when the results don't translate into human clinical outcome. So the question is, how can we use humans as a model system for humans? So as I said, you don't get to perform experiments in humans, but here's something that we can do. Each of us is an experiment of nature. Each of us is an experiment where Mother Nature has perturbed thousands of genes in our genome, and we can measure outcome, clinical outcome of different people to see how that connection between genotype and phenotype manifests. And this is a place where you actually do have a Moore's Law slide. This is a graph again on a logarithmic scale, and this is the number of human genome sequences, the very first human genome in 2001. And you can see that not only is this growing exponentially, it's growing exponentially twice as fast as Moore's Law. So depending on whether you believe this trend line to continue, the number of human genome sequenced by 2025 or 2027 will be somewhere around 100 million to 2 billion. That's a lot of genomes. Genomes on their own are great. When combined with phenotypes, they are better. There's less human clinical outcome data available, but there's more and more coming. One of my favorite resources in this regard is the UK Biobank. The UK government was very forward-thinking in creating a cohort of 500,000 just normal individuals with whatever diseases they may or may not end up developing. And they collected thousands and thousands of phenotypic outcome data on them, including multiple covariates like diet and environment and urinary and blood biomarkers, and for 100,000 of them, full body and brain imaging, and many, many other phenotypes that are incredibly valuable. And there's more of these cohorts emerging. Here in the United States, we have the All of Us cohort, which is about supposed to be 1 million people, also more genetically diverse than the UK cohort, which is predominantly European. So it's going to be a great resource, and there's others emerging over time. So if you take genotype and phenotype and put them together, you basically have a suggestion of causality. And because to a reasonable first cut approximation, a genetic variant that associates with disease is roughly causal of that disease, if you can actually figure out what that is. So that's great, and sure enough, from the in the last 10 or so years since these efforts first started, there's been thousands of traits that have been associated, each with hundreds or thousands of different genetic loci that associate presumably causally with this disease. Some of these are diseases, others are for normal traits like height or even educational attainment or bone mineral density. All of these have genetic loci associated with them. Now, the thing is, for each of those traits, there's usually hundreds of different loci in the genome that have an association with that trait, often with a very small effect size, and how do we figure out which is which? Now, so it turns out that indeed when you have genetic evidence from human genetics for a disease, if you have a drug against that target, it increases your probability of approval. So if you look at the graph on the right-hand side here, you can see that the odds ratio of approval for a drug that has human genetic support is about 2x, which is really an incredible improvement in probability of approval. So that's great. The problem is it's not that simple. Coming back to what I mentioned before, not all targets in this case are created equal. If you look at targets that are Mendelian, in the sense that it's a one gene, one disease association, that odds ratio creeps closer to 3. The ones that come from the genome-wide association studies, where, as I said, there's hundreds of loci and you don't know which one matters, that odds ratio is actually less than 1.5. So it's actually difficult to figure out which of those makes a difference. So what we're going to do here is we're going to come back to this other revolution that I mentioned earlier, where we have high content biological data, and we're going to see if we can get that to get us closer to the causal biology. So let me give you an example of how that might work. This is a case study. It's just a really beautiful case study of psychiatric disease. This is a region on chromosome 16 called 16p11.2 that for whatever reason is subject to copy number alterations in the population relative to wild type. It's deleted in some people and duplicated in others. When it's deleted, there's a 75% probability of autism, and when it's duplicated, a 40% probability of schizophrenia. 25 genes in the region, no one knows which of them has that effect. Up until a paper about 3 years ago from UCSF that took IPS lines from the deletion patients, the duplication patients, and normal controls, reverted them, differentiated them into neurons, and looked at them under a high-content microscope. And what you see is this, and even to the untrained eye, you can see that the neurons on the left look a lot bushier than the neurons in the middle, and the ones in the middle are a lot bushier than the really naked neurons on the right. There's a significant deficit, there's a significant increase of synaptic arborization on the deletion and a significant deficit on the duplication. So we don't know still what's causing, which of the genes in the region causes this, but this gives you a phenotype that you can now try in this IPS culture various interventions to see which of those reverts the phenotype from the unhealthy to the healthy state. So that gives rise basically to what we're doing at In Sitro. Can we do this at scale and using machine learning rather than manual looking at things under the microscope to get at what distinguishes unhealthy versus healthy cells of people with a lower or higher disease burden of genetic disease burden? So if you imagine these are cells that are embedded in a manifold like we talked about in the images side, you can imagine that just like in the image that I showed you before, you have clusters that look like clusters that are healthy that look the same, and you have different types of unhealthy clusters, each of which looks different, and those clusters will emerge by looking at the cluster diagram in the manifold, and we can now ask ourselves which intervention might revert an unhealthy to a healthy cluster. Now, what's important about this is that this manifold gives you two things. It gives you, first and foremost, a stratification of the segmentation of the patients into subtypes that might not be visible at the clinical level. The big transformation that happened in precision oncology was when we realized that breast cancer was not one disease. We were able to do that because we had a lot of molecular data from enough patients that were obtained from tumor biopsy samples of those patients. And we now know that a HER2 positive cancer is very different from a BRCA1 cancer, is very different from a triple neg cancer, and each of those is treated differently today, which is what's given rise to the tremendous advancements in treating cancer, not just for breast cancer, but for other cancers as well. Here, because we will have enough molecular data from enough different genetic backgrounds, these different clusters will emerge. And because we have a cell-based system that is very scalable and intervenable, we can ask what intervention, what drug might allow us to move from, say, the yellow cluster to the blue cluster. So let me give you just a couple of examples to show you that this actually works. This is work that came from our own lab. We've created massive data sets that look at different cells under different conditions with different genetics and so on. And this is just a few examples to show this. This is a bunch of cells, each of which were treated with a CRISPR intervention with targeting a different gene. And the question is, can you, just by looking at the cells, figure out the different genetics of those cells? And the answer is you can't. This is what the manifold looks like, and you can clearly see clusters that emerge. And this is a comparison to the state-of-the-art method that used an engineered set of features. This is a fairly subtle perturbation because it only touches one gene and it only reduces its activity by 20 to 40%. So the fact that you can get this level of performance is quite striking. I'm going to show you the one last one, which is very recent work. This is in a disease that we're working on called NASH, non-alcoholic steatohepatitis. It is a fatty liver disease whose prevalence in the population is becoming increasingly large because of the obesity epidemic and the type 2 diabetes epidemic. And it's going to become the largest cause of liver cancer and liver transplants in the next decade. So the question is, can you create a cellular phenotype for NASH that you can then screen for interventions that revert the phenotype? So this is a bunch of cells from NASH patients and controls, and you ask yourself which are the ones that are NASH and which are the ones that are different. I'm sure you can't tell the difference, but the important thing is neither can a trained cell biologist who studies NASH. And even when we highlight the ones that are NASH versus non-NASH, you kind of look at this and you say, I don't know. And that is exactly what they said. So we've plugged that into a machine learning algorithm to distinguish NASH from non-NASH, and you can see a nice separation in the training set. What's more important is you can see a similar separation in the test set, and rigor of machine learning. This was not only done on a different set of patients, it was done on a different set of patients whose cells were obtained from a different vendor, just to make sure that we're not overfitting. And so this is what you get. And again, you can start to look at the embedding and see what insight that gives you in the biology. You can't tell that much from something of this size. But when you look at the highest ranked tiles that for the NASH versus the highest ranked tiles for the non-NASH, you can clearly see that it's latching onto a phenotype that corresponds to these green circles are lipid droplets. The big pink circle is the nucleus, lipid droplets that are at the nuclear membrane. And you can use attention models from the machine learning to look at what exactly was the network looking at by backpropagating through the network. And you can see that, in fact, in the NASH case, it was looking exactly for those lipid droplets at the nuclear membrane versus the non-NASH was looking at a diffuse signal. So wrapping up, there is what we're really building is an incredible data factory that uses all of those tools that I mentioned before: iPS cells, CRISPR, high content phenotyping, and with automation at scale to create massive amounts of biological data. On the other side, we're using techniques from statistical genetics and from data science and from machine learning to interpret those data, but these are not two separate things. These are two loops that feed into each other. All of the work that's done is done in interdisciplinary teams that work together like this to figure out what is the problem that we can solve together, what is the experiment that we need to perform, what assay do we need to build that really captures the biology, and what model do we build and what insight can we extract from the result. This type of interdisciplinary collaboration, I think, is absolutely critical in this space, and I think in other areas as well. The separation of data scientists into one side of the organization where data are thrown over the wall and results are thrown back over the wall is the wrong way to do data science. So just highlighting that for those of you, I think this is absolutely critical to success. So I'm going to take a big step back now. For the last minute or so of this talk, just mention why I think this is a really wonderful space to be in, this intersection of machine learning / data science with biology and health. If you look back at the history of science, there are, at different periods in history, one discipline that takes on an incredible rate of progress because of a new insight or a new invention or a new way of measuring things. In the 1870s, that discipline was chemistry, where we understood the periodic table and moved away from alchemy and trying to turn lead into gold. In the 1900s, that discipline was physics, understanding the connection between matter and energy and between space and time. In the 1950s, that discipline was computing, where we were able to use silicone chips to actually do calculations that up until that point, only a human, and sometimes not even a human, had been able to do. And then in the 1990s, there was an interesting bifurcation because two disciplines suddenly took on that incredible progress. One was the era of data, which is related to but different to computing. It includes elements of computing, but also of optimization, statistics, and neuroscience. The other era was that of biology, quantitative biology, where we moved away from a purely descriptive science of cataloging phenomena to really understanding the principles of what drives biological systems. And this was enabled by the tools that measure biology in a very quantitative way. Microarrays, the human genome, super resolution microscopy, and so on, all allowed us to really measure biology in unprecedented ways. But those two disciplines didn't talk much to each other. I think the next era that's coming upon us now is what I call digital biology. It's the ability to measure biology in unprecedented scale, detail, and fidelity. The ability to interpret what we measure using the tools of machine learning and data science, and the ability to take that insight and rewrite biology to do things that it wasn't meant to do. And that will have an impact in biomaterials, in agriculture, in energy, in the environment, and in human health. And I think that's an area which is a really exciting place to be, because I think that is the next big epoch of science. Thank you very much.
Beautiful.